Hello students, I am Gaurav Kumar Jain from Jamia Hamdard University, New Delhi. In this module, we will discuss about Preformulation Part 2 under paper Product Development 1. The learning objectives are: number 1, micromeritics and its role in preformulation; second, importance of particle characterization in preformulation; third, importance of density and porosity in preformulation; and fourth, importance of compressibility and flowability in preformulation. Now, let's begin with the introduction part. Micromeritics is the study of properties of small particles. The study of micromeritic properties of particle has implications on number of processes. The first one to study is dissolution. The surface area per unit weight, which is known as specific surface, is increased by reduction in particle size. Increase in surface area by particle size reduction increases the rate of drug dissolution in accordance with Noyes-Whitney equation. The second process which is affected by particle size or micromeritic property is appearance. Feel, texture and color of certain excipients or drugs depends upon the particle size. As an example, the difference in color of red and yellow mercuric oxide is due to difference in their particle size. Particle size may also affect the texture, taste and rheology of oral suspensions. Elegance of emulsion in suspensions often depends upon the particle size of the dispersed phase. The third is flowability. The flow properties of powders are dependent upon the particle size, size distribution as well as particle shape. Asymmetric and small particles have poor flow characteristics and therefore granulation techniques are used to convert powder into granules which provide uniform size having good flow properties. The next property which is affected is compressibility. Physical properties of powders such as compressibility, porosity and bulk density are dependent on the particle size and size distribution. For example, difference in bulk density of light and heavy magnesium carbonate due to difference in their particle size. The next property which is affected is rheology. Particle size influences the duration of adequate serum concentration, rheology and product syringeability of intramuscular injections. The next one is weight uniformity. Weight uniformity of solid oral formulation is also dependent on particle properties. Symmetric spherical particles with good flow and compressibility results in uniform feed from hopper to the die cavity of tableting or capsule filling equipment, allowing uniform particle packing and a constant volume to mass ratio which maintains dose uniformity. The next property which is affected by micromeritic properties is drug release. The release characteristic of drugs from ointments, creams and suppositories are also dependent on the particle size of the dispersed drug. Another property which is influenced by micromeritic property is stability. The stability of biphasic formulations including suspensions and emulsions depend on the particle size, and increase in particle size decreases the stability of these systems based on well-known concept of Stokes' law. The next process which is affected by micromeritic property is mixing. The mixing of several solid ingredients is easier and more uniform if the ingredients are of approximately same size. This provides a greater uniformity of dose. Solid pharmaceuticals that are artificially colored are often milled to distribute the coloring agent to ensure that the mixture is not mottled. The next process which is affected is drying. The drying of wet masses may be facilitated by size reduction, which increases the surface area and reduces the distance that the moisture must travel within the particle to reach the outer surface. During tablets production by wet granulation process, the sieving of the wet mass is done to ensure more rapid and uniform drying. The next process which is affected by micromeritic property is extraction. The particle size reduction during extraction process results in increased surface area and increased area of contact between the solvent and the solid, thus resulting in complete extraction. In the previous module, we have studied about crystallinity, polymorphism, crystal habit and hygroscopicity. Now, we move on to next very important parameter, that is particle size. As discussed above, the study of particle size before product development is important because particle size play important role in processing of materials, appearance of product, dissolution and drug release, and stability of product. In case particle size is difficult to measure, the particle volume or surface area can also be quoted. The size of a spherical particle can be expressed in terms of its diameter. The surface area is proportional to the square of the diameter and the volume is proportional to the cube of the diameter. Thus, for a perfect sphere, the surface area is given by equation S is equals to pi d square and the volume is given by the equation V is equals to pi d cube by 6. However, in naturally occurring particulate solids and milled solids, the shape of particle is irregular with different number of faces. A non-spherical particle has a definite surface area and volume, but being asymmetric, its apparent length varies with its orientation. As the degree of asymmetry increases, however, so does the difficulty of expressing size in terms of meaningful diameter. For this reason, a non-spherical or asymmetric particle is often considered to approximate to a sphere, which can then be characterized by determining its diameter. Because measurement is then based on a hypothetical sphere which represents only an approximation to the true shape of the particle, the dimension is referred to as equivalent spherical diameter of the particle. The size of the particle is expressed in terms of equivalent spherical diameters by using some measurable property such as surface area, volume, diameter or density. Now we will explain these individually. Surface diameter is defined as the diameter of a sphere having the same surface area as that of asymmetric particle in question. Volume diameter is the diameter of a sphere having the same volume as the asymmetric particle in question. Projected diameter is the diameter of a sphere having the same observed area as the asymmetric particle in question when viewed normal to its most stable plane. This diameter is usually determined by microscopic technique. The next diameter to discuss is Stokes diameter, which is referred to as diameter of a sphere with the same density as the asymmetric particle in question and which undergoes sedimentation at the same rate as the asymmetric particle in a given fluid within the range of Stokes' law. This diameter is usually measured by sedimentation method. Unless the particles are asymmetrical in 3 dimensions, these diameters will be independent of particle orientation. The other 2 diameters, the value of which are dependent on both the orientation and the shape of the particles, are Ferret's and Martin's diameter. Ferret's diameter is defined as the mean distance between 2 tangents on opposite sides of the particle parallel to some fixed direction. Martin's diameter, on the other hand, is the length of the line that bisects the particle. The line may be drawn in any direction but must be in the same direction for all the particles to be measured. A particle population which consists of spheres or equivalent spheres with uniform dimension is monosize and its characteristics can be described by a single diameter or equivalent diameter. However, most pharmaceutical powders are polydisperse, that is, they consist of a mixture of particles of varying sizes and shape. It's therefore become necessary to know not only the size of the particle in the sample but also the number of particles of each size present in the sample. This is called as particle size distribution. Thus, we need an estimate of the size range present and the number of particles in each particle size, and from this we can derive the average particle size of the collection of particles given by the following equation. Important to understand, in the above calculation, only the number and mean size of particles have been considered for expressing the average particle size. Certain modifications in the calculation can be done in order to take into account the surface and volume of the particle. Such modified equation for calculation of average particle size is termed as Edmondson equation, given by the following. In this equation, n is the number of particles in each size range, d is the diameter of particles in a given size range, p is an index related to size of an individual particle, and f is the frequency index. Value of P can be 1, 2 and 3. When P is equals to 1, it indicates particle length. When P is equals to 2, it indicates particle surface. When P is equals to 3, it indicates particle volume. Value of F can be 0, 1, 2 and 3. F0 express size distribution in total number. F value of 1 express size distribution in length. When F is equals to 2, it expresses size distribution in surface, and F value of 3 express size distribution in volume. The evaluation of such modified diameters play a very important role, particularly in the development of pulmonary dosage forms such as DPIs and MDIs, since the size of the particles based on its volume and surface area is useful in determining amount and pattern of lung deposition of the drugs. Particle size and size distribution are determined by several methods available, first of which is optical microscopy. This method is based on counting the particles by visualizing them in suspended form under microscope using calibrated scale. This method is suitable for determining size of the particles ranging from 1 to about 100 microns. In case particles are smaller than this range, then electron microscopy techniques such as scanning electron microscope or transmission electron microscope are used. By the microscopy techniques, projected area diameter, Farrett's diameter and Martin's diameter can be determined. The another technique which is used for determination of particle size and size distribution is sieving. This method is based on determining weight of particles retained on the sieves arranged in increasing sieve number. This method is suitable for determining size of the particles ranging from 5 microns to about 1000 microns. The next technique is sedimentation technique. This method utilizes Anderson pipette apparatus. The sedimentation method is based on the dependence of the sedimentation rate of the particles on their size as expressed by Stokes' equation given below. This method is suitable for determining size of the particles ranging from 1 microns to about 200 microns. The next technique is photon correlation spectroscopy. This technique is widely used to determine size of particles based on scattering of light by the suspended particles. Technique can be best utilized by particles in the size range of 10 nanometers to 5 microns. The last technique for determining particle size, size distribution and also for particle volume is Coulter counter. It operates on the principle that when a particle suspended in a conducting liquid passes through a small orifice on either side of which are electrodes, a change in electrical resistance occurs. The change in electrical resistance is proportional to the volume of the particle. The range of analysis of Coulter counter varies from 0.1 microns to about 1000 microns. This is very fast and reliable technique for size and size distribution determination. The next particle characteristic is surface area. The surface area of drug particles are important because dissolution is a function of this parameter, as described by Noyes-Whitney equation. Surface area can also be quoted if the particle size is difficult to measure, as previously told. The precise measurement of surface area is made by Bronner, Emmett and Teller, that is called as BET equation. It involves nitrogen adsorption, in which the layer of nitrogen molecules is adsorbed to the sample surface at minus 196 degrees Celsius. Once surface adsorption has reached equilibrium, the sample is heated to room temperature, the nitrogen gas is desorbed and its volume is measured and converted to the number of adsorbed molecules via the ideal gas law. Since each nitrogen molecule occupies an area of 16 A square, one may readily compute the surface area per gram for each pre-weighted sample. The method is commonly called as quanta-sorb. Another method based on permeability of air, called as Fischer-Subsieve sizer, is also used for determination of surface area of powder particles. This method is based on the principle that the resistance offered to the flow of a fluid such as air through a plug of compacted powder is proportional to the surface area of the powder. The greater the surface area per gram of the powder, the greater is the resistance to flow. Next characteristic is particle shape. Particle size combined with particle shape affects the packing properties and the flow of a powder, and it also has some influence on the surface area. It is generally accepted that the flowability of powder decreases as the shape of the particle becomes more irregular. The first factor to describe is sphericity. Sphericity is a measure of the roundness of a shape and is independent of particle size. The sphericity of a particle is the ratio of surface area of a sphere to the surface area of the particle, where sphere has same volume as that of particle. Sphericity is a ratio and therefore a dimensionless number and is calculated for any three-dimensional object if its surface area and volume are known. Sphericity value of some common shapes are shown in table. Tetrahedron has sphericity value of 0.671. Cube has sphericity value of 0.806, octahedron has sphericity value of 0.846, cone shape has sphericity value of 0.724, cylindrical shape has sphericity value of 0.874 and sphere has sphericity value of 1. The next factor is elongation. Elongation provides an indication of the length is to width ratio of the particle and is defined as 1 minus width upon length. Shapes symmetrical in all axes such as circles or squares will have an elongation close to 0, whereas needle shaped particles will have value closer to 1. Convexity. Convexity is a measurement of surface roughness of a particle and is calculated by dividing the particle area by a total area. An irregular or spiky shape has a convexity closer to 0, while a smooth shape has a convexity of 1. The last parameter to describe particle shape is circularity. Circularity is a measurement of the ratio of the actual perimeter of the particle to the perimeter of a circle of the same area. An irregular or spiky shape has a circularity closer to 0, while a perfect circle has a circularity of 1. The next property to study is porosity. Porosity is a derived property which is dependent upon the fundamental properties which we have studied till now. Porosity is a measure of the air spaces or voids in a material. In a powder bed, three types of air spaces or voids can be distinguished. Number 1, open intraparticular voids are those within a single particle but open to the external environment. Closed intraparticular voids are those which are present within a single particle but close to the external environment. Interparticulate voids are the air spaces between the individual particles. Based on the type of voids, three interpretations of powder volume are there. Number 1, bulk volume, which is defined as total volume occupied by the entire powder mass including voids. Number 2, granular volume. This is volume of solid particles exclusive of interparticulate but not intraparticulate void spaces. The third one is true volume. This is volume of solid particles exclusive of both inter and intraparticulate voids. The ratio of total volume of void spaces to the bulk volume of the material is often referred to as porosity of the material. Porosity is frequently expressed as a percentage in form of the equation mentioned below. The next derived property is density. Density can be defined as ratio of mass of an object to its volume. Therefore, the density of a solid is a reflection of the arrangement of molecules in a solid. Based on the type of volume defined, corresponding density may be proposed. The bulk density of a powder is obtained by dividing its mass by the bulk volume it occupies. The bulk volume is the volume of the powder as poured or as passively filled into a measuring vessel and includes both inter and intraparticulate spaces between and of the particles. The bulk density is often very difficult to measure since the slightest disturbance of the powder bed may result in new bulk density. The density corresponding to granular volume is termed as granular density. The true density of a material is the density of the actual solid material. Unlike true density, bulk density of a powder is not a definite number but an indirect measurement of a number of factors including particle size and size distribution, particle shape and the method of measurement. The bulk density of a powder is always less than the true density of its component particles because of the presence of pores or voids. This statement reveals that whereas a powder can only possess a single true density, it can have many different bulk densities depending on the way in which the particles are packed and the bed porosity. Another density term, that is tap density, also called as compressed bulk density, is the limiting density of a powder attained after compaction by tapping or vibration following a specified procedure. Another density term, that is tap density, also called as compressed bulk density, is the limiting density of a powder attained after compaction by tapping or vibration following a specified procedure. The sample is usually tapped or vibrated until an equilibrium volume is obtained, and at that point the final tap density is determined. The various types of densities are summarized below. The first type, as already mentioned, is bulk density. It is defined as mass divided by bulk volume. It is determined by bulk density apparatus or pycnometer. It is characteristic of a powder and it is dependent on particle packing as the powder consolidates. The next density is granular density, which is defined as mass divided by granular volume. It is determined by mercury displacement method. The next density is true density, which is defined as mass divided by true volume. It is determined by helium displacement method. It is characteristic of the particle. The last density is tap density, which is defined as mass divided by volume obtained by compacting bulk volume by tapping. Mechanical tapping device such as Zolting volumeter is used for its determination. This type of density is used to characterize powder flow. In pharmaceutical development terms, knowledge of the density of powders has been used for the determination of the consolidated behavior. For example, the well-known Haeckel's equation requires knowledge of the true density of the compound as mentioned below. In this equation, D is the relative density, which is ratio of the apparent density to the true density. K is determined from the linear plot of the Haeckel's plot and P is the pressure. Information about the density of a powder can be used to predict whether a compound will cream or sediment in a meter dose inhaler formulation. Below we mention few examples to discuss the importance of density in product development. Information about the density of a powder can be used to predict whether a compound will sediment in a meter dose inhaler formulation. The density of the hydrofluoroalkane propellant 134A is 1.217 gram per centimeter cube. Therefore, suspensions of compounds that have a true density less than these figures will cream, that is rise to the surface, and those that are denser will sediment. Those that match the density of propellant will stay in suspension for a longer period of time. Next derived property to discuss is flow and compressibility. Although at the pre-formulation stage only limited quantity of candidate drugs are available, any data generated on flow and compaction properties can be of great use to the formulation scientist. The data provided can give guidance on the selection of the excipients to use, the formulation type and the manufacturing process to use. It is important that once the habit and size distribution of the test compound have been determined, the flow and compaction properties are evaluated if the intended dosage form is a solid dosage form. The importance of solid handling properties, especially flow properties, cannot be overemphasized since solid dosage forms are the most predominant in terms of volume and value. Given below are the few points to mention the importance of flow behavior. First, the flow properties of solids have great impact on the tableting processes since their manufacturing requires the flow of powder from hopper to tablet dies. Second, weight and content uniformity is also dependent on flow of powders. Third, the flow properties of solids also have great influence on the mixing and demixing of powders. Fourth, the speed of tablet production is also greatly affected by the formulation flow characteristics. Finally, for the final product, weight, content uniformity, hardness, disintegration and dissolution are all affected by formulation flow. Powders are probably the least predictable of all materials in relation to flowability because of the large number of factors can change their rheological properties. Physical characteristics of the particles like size, shape, angularity, surface texture, porosity and hardness will all affect flow properties. External factors such as humidity, conveying environment, vibration and perhaps most importantly aeration will compound the problem. Another characteristic of the powder is that they are often inherently unstable in relation to their flow performance and even a free flowing material could cease to flow. This transition may be initiated by the formation of a bridge, floccules, by adhesion to surfaces or by an event that may promote compaction of the powder. The tendency to switch in this way vary greatly from one powder to another but can even be pronounced between batches of the same material. The flow properties of a material result from forces that can act between solid particles including frictional forces, surface tension forces, mechanical forces caused by interlocking of particles of irregular shape, electrostatic forces and cohesive van der Waals forces. All of these forces can affect flow properties of a solid. Most flow properties are significantly affected by changes in particle size, density, shape, electrostatic charge and absorbed moisture which may arise from processing or formulation. In general, powders with large particles greater than 100 microns will be non-cohesive, permeable and will probably fluidize and will have low compressibility and relatively low shear strength. Conversely, fine powders less than 10 microns are likely to be cohesive, compressible, contains much entrained air and yet have poor aeration characteristics. Generally, they have high shear strength, high flow energy, low permeability and are very affected by being consolidated when entrained air is excluded. However, under forced flow conditions, fine powders can behave more like a fluid. They are able to extrude round corners or through holes, unlike coarse powders that are more likely to become solid-like as particles realign and lock together and become very resistant to flow. Next, we will discuss about characterization of powder flow. Commonly used methods for characterizing powder flow are: first, compressibility index, second, Hausner's ratio and third, angle of repose. First, we discuss about compressibility index. Compressibility index is defined by Kars. Kars reported that more a material is compacted in a compaction or tapped bulk density test, the poorer its flow property. A simple indication of the ease with which a material can be induced to flow is given by application of compressibility index as well as by Hausner's ratio given by the following equations. Scale of flowability is discussed below. Flow is considered to be excellent if Kars index values vary from 5 to 15 and Hausner's ratio varies from 1.05 to 1.18. Flow is considered to be good if Kars index value is from 12 to 16 and Hausner's value is from 1.14 to 1.20. Flow is considered to be fair or passable if Kars index value is from 18 to 21 and Hausner's value is from 1.22 to 1.26. Flow is considered poor if Kars value is from 23 to 35 and Hausner's ratio is from 1.30 to 1.54. It is considered as very poor if Kars index value is from 33 to 38 and Hausner's value is from 1.50 to 1.61, and it is considered as very poor if Kars index value is greater than 40 and Hausner's value is greater than 1.67. Compressibility and flow property of some common pharmaceutical excipients are described in the table. The next parameter to discuss is angle of repose. Now, if a powder is allowed to flow onto a flat surface, a pile or heap of powder is formed. This is very well known phenomena. A material that is not cohesive and flows well is spread out, forming a low heap. More cohesive material forms higher heap which are less spreaded out. The angle of repose, that is theta, is defined as angle of the free surface of the pile of the powder to the horizontal plane and is represented by the equation tan theta is equals to h divided by r, where h is height of pile, r is radius of pile and theta is angle of repose. It is the maximum angle that can be obtained between the freestanding surface of a powder heap and the horizontal plane. Such measurements give at least a qualitative assessment of the internal cohesive and frictional effects under low levels of external loading as might apply in powder mixing or in tablet dye or capsule shell filling operations. There are two methods to determine angle of repose: static method and dynamic method. Static angle of repose has two procedures: the fixed funnel method and fixed cone method. The fixed funnel method employs a funnel that is secured with its tip to a given height h above graph paper that is placed on a flat horizontal surface. Powder or granule is carefully poured through the funnel until the apex of the conical pile just touches the tip of the funnel. The radius of the base of the conical pile is then determined to calculate the angle of repose. The fixed cone method establishes the radius of the cone base r by using a circular dish with sharp edges. Powder is poured onto the center of the dish from a funnel that can be raised vertically until a maximum cone height h is obtained. The angle of repose is calculated as before. Dynamic angle of repose, also called as kinetic angle of repose, are preferred since they most closely mimic the manufacturing situation in which the powder is in motion. The first method under this category is tilting box method. In this method, a sandpaper lined rectangular box is filled with the powder and carefully tilted until the contents begin to slide. The maximum angle that the plane of powder makes with the horizontal surface on rotation is taken as angle of repose. The second method is rotating cylinder method. A typical dynamic test involves a hollow cylinder half filled with the test powder with one end sealed by a transparent plate. The cylinder is rotated about its horizontal axis until the powder surface cascades. Now we discuss the importance of value of theta in terms of flow behavior. Theta value less than 20 exists rarely. Theta value from 25 to 30 indicates excellent flow properties. Theta value between 31 to 35 indicate good flow. Theta value from 36 to 40 indicate fair flow behavior. Theta values from 41 to 45 degrees indicates passable flow. Values from 46 to 55 degree indicates poor flow and such powders require agitation. Values ranging from 56 to 65 degree indicates very poor flow. And in the last, theta values greater than or equal to 65 degree indicates very poor flow. As mentioned previously, from the angle of repose and compressibility values, a reasonable indication of material's inherent flow properties could be possible. In this module, we have discussed about the impact of micrometallic properties to preformulation studies. Conclusion of this module is that the micrometallic properties such as particle size, shape, surface area, density and flowability of drug can be related in a significant way to the physical, chemical and pharmacological properties of a drug. The particle properties of the drug can not only affect its release from dosage form but also affects the quality of tablets, capsules, suspensions and emulsions from the viewpoint of both uniformity and stability. Thus, determination of these properties during preformulation study is requisite.